Welcome to Medical Genetics. In Unit 1, we'll go through a brief history of genetics and then look at the molecular biology of DNA. So these are chapters 1 and 2 in our textbook. Let's move on. Chapter 1 of your textbook gives you a brief background in terms of what medical genetics is, how it developed, its history, and as a result I couldn't resist putting on this page a picture of the father of genetics, Gregor Mendel, and then of course the sign for the Human Genomics Project that you see over on the right. When I was a graduate student, we thought that medical genetics or genetics had a significant role in disease processes, but, only maybe 40, maybe 50%. But now we know it has a central role in understanding all major diseases, and this includes those that we think of typically when we think of genetic diseases, the pediatric inherited diseases such as cystic fibrosis, sickle cell anemia, hemophilia, type 1 diabetes, as you see here on the slide. But it also involves common adult diseases such as heart disease, type 2 diabetes, cancer, and many psychiatric disorders. So we now know that medical genetics has a role in just about all major diseases. Medical genetics is defined as any application of genetics to a medical practice. So these applications can be studies into the role of inheritance in families, mapping of disease genes, finding out where the genes are located on which chromosomes, looking at the molecular mechanisms by which genes can cause disease, diagnosis, and of course treatment. There are a number of different gene therapies that are in clinical trials, and these will, I think, increase only with time. And then genetic counseling, where we provide patients and their families the information on risks of passing a genetic disease to offspring, or the prognosis if they have been diagnosed with it, and what the treatment options are. So, how important is medical genetics for your practice? Well, since genetics disease makes up a large proportion of the total disease burden in both pediatric and adult populations, it's getting to be very important. And understanding the genetic mechanism of disease then means we can target more specific therapies to perhaps actually prevent disease development. So let's go a little bit into the history, starting with Gregor Johann Mendel. He is an Austrian monk, or was an Austrian monk, I should say, considered the father of genetics. And of course, I couldn't resist putting a little picture of the peas over here on the lower left, because he figured these out in his garden where he grew some peas. And he formulated a series of fundamental principles of heredity just from observing the different characteristics of the pea plants. Unfortunately, he published his results in 1865 in a very obscure scientific journal, so he received absolutely no recognition for his work for about 35 years. It just goes to show you never get any respect. Now, while Gregor Mendel was working on his pea plants, a lot of other significant things was going on in the world of science in the mid-1860s. It was kind of a heady time. First you had Charles Darwin, of course, getting everybody upset with his theories of evolution. Then you had Francis Galton, who happened to actually be Darwin's cousin, who performed a series of studies on identical twins, again looking at inheritance patterns. Louis Pasteur, meanwhile, was disproving the theory of spontaneous generation, in other words, that living matter comes from non-living matter, and helping to help man understand the nature of infectious disease by accepting the germ theory of disease, that each different infectious disease is caused by a different microorganism. And then you had Robert Koch, the German, who actually established the germ theory of disease and how to go about identifying which microorganism was causing which disease. Robert Koch actually identified the microorganism that causes tuberculosis, and for that he received one of the first Nobel Prizes in medicine. Mendel's principles were actually independently rediscovered in 1900 by 3 different scientists. I don't expect you to memorize their names. You won't be tested on them. You may have heard, however, of Karl Landsteiner, who in 1900 discovered the ABO blood groups, for which he received a Nobel Prize in medicine. In 1902, Archibald Garrod described alkaptonuria, one of the first inborn errors of metabolism. The children are missing a very important enzyme. And in 1909, Johansen coined the term gene as the basic unit of heredity. So several decades later, studies started focusing on the genes and everything using 2 models that turned out to be quite useful. Fruit flies, for one, Drosophila, and the bread mold, Neurospora. And both of them are eukaryotes. In other words, they have cells with membrane-bound organelles like we do. So we could use these models for looking at how our genes are arranged in our cells. Now, the first half of the 20th century, there were some very significant people who helped develop population genetics. This is Ronald Fisher, J.B.S. Haldane, and Sewell Wright. I don't expect you to memorize them, but there were numbers of people working on this. And then modes of inheritance of several important genetic diseases were determined. So phenylketonuria, sickle cell, Huntington, and cystic fibrosis. We began to understand the mechanisms of inheritance of those. And then in 1944, while World War II was still going on, 3 scientists, Avery, MacLeod, and McCarty, proved that genes are composed of DNA. So 1865 is when Mendel is figuring out the basic patterns of inheritance, but it wasn't until 1944 that the genes were located in DNA. So it took a while. The second half of the 20th century was when we really started to understand how the information for proteins and genes are encoded in the DNA, and so we think of the second half of the 20th century as being the molecular biology portion. Remember, it took us until 1944 to determine that DNA carried the genes. Then, just a few years later, 9 years later, Watson and Crick deduced the structure of DNA using Rosalind Franklin's work. 1956, it took that long to determine the correct number of human chromosomes. In other words, we have 23 pairs, or a total of 46 in every nucleated cell in the human body. In 1963, Nuremberg, Matthew, Leader, and Corona deciphered the genetic code. 1975, Frederick Sanger came up with developing the dideoxy method for sequencing DNA. Now, again, don't worry, I'm not going to ask you who did what on this, but notice how the times in between are getting shorter and shorter for significant developments. 75, also a man by the name of Ian Southern developed a technique for identifying the genes. And then in 1983, Kary Mullis developed the polymerase chain reaction for amplifying small amounts of DNA. So going from just barely knowing the genetic code in 1963 to a way to actually amplify small amounts of it in 1983, 20 short years is incredible. Now, in the Human Genome Congress, so we're in 1983, 7 years later, in 1990, the Human Genome Project starts. And they figured that it would take us like 15 years or something, more than 15 or 20 years, and we did it in about 13. And the reason for why we thought it would take so long, because we have 3 billion base pairs. Now, I want you to think of a base pair as being like a single letter. So there were 3 billion letters that we had to sequence in order to get this. So it was quite an effort. Why bother? Well, it's helped us pinpoint molecular defects in many important genetic diseases, and it has given us a better estimate of just how many genes a human has, which surprisingly is as low as 20 to 25,000. Originally, when I was in graduate school, we thought it was about 100,000, but now we find it's a lot less, and we know why that is too. Couldn't resist putting in a little cartoon at this point where we look at Edna's genome research. This is not the kind of genome research that I'm going to be talking about, but I thought it was kind of cute, especially since my sister-in-law loves these garden gnomes. Go figure. So, what kinds of genetic diseases are there? Well, some diseases are a result of chromosomal disorders. You may have too many or too few, or some of them are broken or duplicated or even completely missing. Then you have single gene disorders. The chromosomal disorders, of course, are usually pretty severe because you're missing a fair number of genes, whereas with the single gene disorder, it's just the sequence of one gene has been altered and then affecting the function of that gene. Multifactorial disorders, this is where genetics and environmental factors will have an effect. And mitochondrial disorders, we're beginning to discover more and more of these. And even though it says there's a small number, they're becoming, as we understand them, a little bit more prevalent than we originally thought. And what you may not have remembered is that mitochondria have their own chromosome, not nearly as complex as ours, but if there's a mutation in those, then it can affect our ability to make ATP. So just to kind of blow you away and tell you relax, you're not going to have to know all of these diseases by any manner of means and their prevalence, how often they occur, but this just shows you some of the chromosomal abnormalities, the single gene disorders, multifactorial, cancer, all types, schizophrenia, and the mitochondrial, all are included in this. So, what about the single gene disorders? They're the ones that Mendel was looking at when his pea plants had independent assortment of different characteristics. So they're classified according to the manner in which they are inherited in families. They can be autosomal dominant, autosomal recessive, or X-linked, or even Y-linked, although that's kind of rare. But you should be autosomal, where if you don't remember that, it's important that you get that one cleared up early on, and we're going to do that in Chapter 2 in just a short bit. And then I couldn't resist talking about Victor McCusick, who started a volume called The Mendelian Inheritance in Man about the single gene disorders. He started publishing it in 1966. And at that time, there were only 1,300 autosomal traits, maybe 119 X-linked traits. Well, on his new online version, there are almost 15,000 autosomal, 893 X-linked, 56 Y-linked, and 64 mitochondrial genetic disorders that have been identified and characterized enough to be included. I couldn't resist giving you a picture of Victor McCusick. I had the great privilege of meeting him at a meeting some time ago. We don't have to talk about it. He actually recently passed away just a few years back. But he's a fascinating man. And it turns out he got interested in genetics because he was an identical twin. And his identical twin was just as intelligent and was almost appointed to the Supreme Court. So it was quite a family and quite an honor to have gotten a chance to meet him. Well, genetic disorders then may result from single gene conditions, multiple gene interactions, or multiple genes and environmental interactions, which make them then very complicated. Therefore, what you need to start thinking about is that genetic disorders lie along a continuum. you have disorders that are environmentally determined. This, in many cases, is a result of an infectious disease, okay, or exposure to a poison. But at the same time, at the other end, you have diseases that are caused because of a mutation in a gene or chromosomal disorders, etc., and that would be the other far extreme. But most disorders are somewhere in between: heart disease, diabetes, birth defects, cancer. Why haven't we solved all of these and know how best to treat it? Is because genetic factors and environmental factors both contribute to the development of the more common disorders. So let's think about that clinical impact of genetic disease again. In 1900, the majority of the diseases were caused by environmental conditions. We died of infection most of the time, or malnutrition, or unsanitary conditions which led to infection. In 2008, genetic diseases account for an increasing proportion. In fact, now we estimate that 3 to 7% of the population in the industrialized nations now will be diagnosed with a genetic disease. Why? Because we now have the means of detecting them, whereas in the past we didn't. So if you take a look at 1900 here, you're not going to have to memorize this, but you can see number one killer was influenza pneumonia, followed only shortly a little bit by tuberculosis, gastroenteritis. Then you had heart disease and stroke, and infectious diseases, and look at diphtheria. But now, 2008, heart disease, cancer, stroke, pulmonary disease, accidents, and now we get down to the infectious diseases. So things have really flip-flopped a great deal. But that might not stay that way if antibiotic resistance and stuff keeps going on So let's look at the approximate prevalence of genetic disease in the general population. Lifetime prevalence per 1,000, an autosomal dominant trait. In other words, it's seen in the adult and seen in the offspring, anywhere from 3 to 9.5 per 1,000. Autosomal recessive, less likely, 2 to 2.5. X-linked, even less, but up to 2%. If we start looking at chromosomal disorders, we can go 6 to 9 per 1,000, I should say. Look at congenital malformation, which has way too many R's in there, but it's anywhere from 20 to 50. So that gives us a total of, 31.5 to 73 people per 1,000, where genetics has a direct effect on their disease. Okay, and then you start to think about how genetics will influence the more common diseases, and you can see that genetics plays a